Hi, everyone. Welcome to episode 109 of Cell and Gene, the podcast. I'm your host, Erin Harris. Today, I'm joined by Dr. Will Chou, CEO of Passage Bio. And Will and I get into a deep dive into the promise and the real world challenges of developing gene therapies for frontotemporal dementia, or FTD. We get into what makes FTD such a complex disease to tackle, from its genetic roots to the hurdles of targeting the brain. Will walks me through the latest interim results from their upliFT-D study of PBFT02, including safety and tolerability so far, and how different dose cohorts are stacking up and evolving strategies for immunosuppression. We also talk about what's next for PBFT02 and what these milestones could mean for the future of gene therapy in neurodegenerative diseases. Neurodegenerative diseases are gaining more and more traction and coverage in our space, and so this is a really important episode to check out. And so with that, enjoy my conversation with Passage Bio's Dr. Will Chou. Enjoy. Will, hi, welcome to Cell and Gene, the podcast. I'm so happy you're here. I've had the pleasure of working with Passage Bio and the various professionals who work throughout the company, but I don't think I've had yet the opportunity to talk directly with you. So this is a pleasure. Thanks for being here.
Thank you for having me, Erin.
Good. So talk to us about Passage Bio for our listeners who might not be completely familiar with the company, as well as FTD and the causes at what is causing it at a genetic level.
Sure. So Passage Bio is developing genetic medicines for neurodegenerative disease. And as you mentioned, our lead program is in the clinic in a phase 1/2 study for frontotemporal dementia. Now, FTD, as it's called, is the most common early onset form of dementia, and it's a little bit different from Alzheimer's in that it's more of a behavioral dementia where patients, they do have memory loss, but they have issues with word finding, communication, and then changes in their personality. So it's a very difficult disease for patients. There are currently no therapies for FTD. And about 30% of FTD is caused from a genetic cause. And of those genetic causes, about a third of them are from what we are studying, which is FTD with the granulin mutation. And so we are looking at a very focused set of patients who have a specific mutation that causes their FTD.
Great. Okay. And we'll definitely get into the specifics as we go on in our conversation. But before we kind of take a deep dive into FTD from Passage Bio's perspective, I'd like to pick your brain about, what are perhaps the biggest scientific, manufacturing, or regulatory hurdles that are facing the development of gene therapies for FTD since, we don't have a cure?
Yeah, absolutely. So I would say from a scientific point of view, it piggybacks on to what I mentioned before. So first, if you talk to FTD patients and you go to the FTD patient community, there's the vast majority of patients with FTD do not have a genetic form. So that's one big scientific issue, is that we don't have a good way of treating or even targeting the non-genetic forms of FTD. And then even within genetic FTD, there's three main mutations. There's the granulin mutation, there is the MAPT mutation, and there's the C9orf72 mutation. And the vast majority of research right now is doing what we're doing, which is going after the granulin mutation, because this is a mutation where the patients have that mutation, they're deficient in a protein, you can replace that protein and try and reverse the damage, right? So it's very straightforward gene replacement therapy. The other forms, genetic forms of FTD, there's not as direct of a correlation between the gene deficiency and the actual pathology that delivers the FTD. So it's not as straightforward of an approach. And we know that genetic FTD patients are a little frustrated in that if you have the granule mutation, there's 4 different programs that are chasing this. other mutations, there aren't as many. And so that's a big scientific issue that we know the community is trying to address for the patients. From a manufacturing standpoint, I'm going to keep this really simple. In our own experience with manufacturing our gene therapy, we've made fantastic progress. And so we can make this gene therapy at scale. One relatively small batch could treat over 1,000 patients. So we've got the manufacturing down quite well. And it's one of the advantages of doing an AAV therapy. So we are doing AAV. It's a rather old school. I will put that in quotes. It's a older technology, but one good thing about having a technology that's been around for a while is there's a lot of experience in how to manufacture it. And so we've been very quickly been able to get to a very efficient way of manufacturing this product. From a regulatory standpoint, I will say the question that we get from most people is, what have you experienced with the new administration from a regulatory standpoint? And from our own experiences, the turnaround time has been very fast. And from what we've been hearing, both in the public, and I was at the FDA a couple of months ago speaking with Dr. McCary, Dr. Prasad, they want to continue in the path that has been set before them at CBER, which was they want to give patients the opportunity to try therapies. That was very important. And Dr. McCary emphasized that he believes in both the words and the action of right to try. And so they understand that for rare diseases, which is what we are dealing with here, you're sometimes going to need alternative ways of experimenting because you don't have as many patients. And so you're going to need some creativity with your analysis plans. And we believe that this administration and this FDA is going to continue with that great flexibility and understanding that the previous administration had.
Good. That's great news. And certainly promising, especially from a regulatory standpoint. Real quick on the manufacturing, are you, let our listeners know, are you manufacturing the gene therapy in-house? Or are you working with the CDMO? Is there some sort of hybrid partnership?
Yeah, it's great. So great question. So we are working with the CDMO. We did all of our development in-house, and so developing what the new process is going to be. But the actual process we're doing at a CDMO, we work with Catalent, and we've had great success with Catalent in making the product both with the old method and the new method. And I think this is a big misunderstanding that is out there. We talk about, so we're a small biotech, we talk about larger partners, and there are some partners who are interested in gene therapies and some who are not interested in gene therapies. And one thing we hear a lot is, oh, well, there's all of this manufacturing investment that needs to be made if you want to be in gene therapies. And what I would say is that's not necessarily true. There's there is ample supply out there right now with CDMOs for manufacturing supply. So there is, with some of the contraction in the market, there's actually now a lot of available supply for manufacturing. And in the case of programs like ours, if you have a manufacturing process developed and it is very inexpensive to do at scale, there is no need for any company to invest in all of the in-house manufacturing. And because when you do that, you have to fill that manufacturing, right? You have to have other products that will fill that capacity. And then you have a constant pressure of, oh, I need more and more genetic medicines. Well, you don't necessarily need to have that model. I think the CDMO model is absolutely doable. There's ample supply, and you shouldn't need to have to make a major investment in a manufacturing plant to be able to do genetic medicines.
Yeah, makes perfect sense.
Yep.
Okay, I want to talk a little bit about the latest interim data for
Sure.
PBFT02 that was presented at AAIC 2025. Can you bring us up to speed on that, especially for people who maybe did not attend?
Sure, absolutely. So What we're providing is an update on our ongoing phase 1/2 study. And so some of the key highlights are first, a continued very high durability of response. So the protein that this gene therapy PBFT02 is making is progranulin, and we are measuring progranulin levels in the cerebrospinal fluid because that is the target, and that's our measure of target engagement. And what we're looking to find is high levels and lasting over time. And the data we've shared at AAIC is really a continuation of what we've shown before, that through 18 months, we've shown very high and very durable CSF progranulin responses. So our target engagement is high. There have been other genetic medicines in this space that have shown a decline in CSF progranulin after a single dose of an AAV. And that's what we want to show does not happen with our product. If you're getting a gene therapy, you do want the effect to be permanent. You want it to be a one-time administration. And so very good signs there for durability. The second thing I'll say is that the product continues to show a slowing of neurodegeneration. And we measure that by looking at plasma neurofilament levels. So plasma neurofilaments are the only biomarker in FTD GRN where we understand what the natural history looks like. And so in the natural history data, and really we replicated some natural history data with our own analysis. In both of these analyses, it's about a 29% annual increase in plasma neurofilaments in patients who are untreated. Just your normal expected FTD-GRN patients, that's the increase you would expect. And our patients so far after a year are at about a 4% increase. So a real separation between natural history and what we're seeing in our treated patients, which is what we want to see, that the product is slowing neurodegeneration. And then the final new piece of data that we shared is have started dosing patients with a lower dose because we saw such a robust progranulin response with our first dose. We made the decision to test a lower dose. And that lower dose, we only have one data point that we're sharing from it. It's very early on, but it looks very promising because it's already at near the upper limits of the normal range. If you look at what the pattern of increase in progranulin has been for our other patients, based on this early signal, we feel very good about the prospects for this lower dose.
Good, good. Okay. One thing that I wanted to ask you up top, and I think it's important that maybe we cover it here. Is there an age range of onset for or sign of symptom for people with FTD? I know we talked about, like we said, symptoms, but not necessarily age range. Is that part of the equation?
Well, the age range is variable, so it's anywhere from 40 to 60 on average for
Mm-hmm.
first onset. But, you know, what we're looking at for when we look at patient attributes is where they are in their disease process, not necessarily their age.
Sure. Okay. Fair enough. I want to talk a little bit about safety and tolerability of
Sure.
PBFT02 from the upliFT-D study. First of all, if you could talk to us a little bit about what the upliFT-D study is, and then talk to us about any insights about immunosuppression management and adverse events that may have influenced protocol adjustments.
Sure, sure, sure. So the upliFT study, that is the name, that's the official name of our ongoing phase 1/2 study. And so let me talk about immunosuppression first. So this product, PBFT02, is administered directly to the CSF. We're not administering it peripherally. And so the doses that we need to use are much lower than, say, the doses of, say, the Sarepta product that are given. So our doses are in the E to 13th range. So 2.2 times 10 to the 13th total genome copies are administered. That's 1 to 2 logs lower than the systemic AAVs. And what we've seen is that There is some spillover. We would expect some spillover into a systemic response, but the amount of immunosuppression that we need is not nearly as high as some of the systemic therapies. So we are at an immunosuppression regimen of 3 days of pulse IV steroids, followed by oral steroids for a 2-month course. So all we're giving is steroids for immunosuppression. And based on our experience so far, that's all we're going to do for immunosuppression. That is all we need for immunosuppression.
Sure.
Here and there, we've had one patient who had a breakthrough with higher LFTs. They responded to more steroids. We're not going to add anything else for immunosuppression because this is a relatively low dose that we're giving. Now, one thing that we have discovered, we've had a couple of patients who relatively early in the course had an SAE known as a venous thromboembolic event. So a venous sinus thrombosis is a blood clot in the venous sinus, which is the first place where CSF drains in the brain. These were asymptomatic. Often these events are not discovered at all in patients. They just kind of happen and you just find them on MRI. But because of the proximity to the product, it is possible that the product could be causing a local immune response that's leading to a hypercoagulable state that's leading to these venous sinus thromboses that happened in 2 patients. Now, even though these patients were asymptomatic and it completely resolved with anticoagulation, what we have decided to do is moving forward, all patients who are treated with our product are going to be administered low-dose prophylactic anticoagulation for a few weeks after getting this product. And the reason we've decided to do this is we don't want to add any additional risk. And we've seen this event in 2 patients. It's kind of an unusual event, and it is pretty proximal to the product. So even though we can't 100% say this is from the product, we don't want to introduce that risk. Low-dose anticoagulation has been broadly studied in thousands and thousands of older patients who are getting hip replacements or knee replacements. And so if you were to go for a knee replacement, you would be on oral anticoagulation while you can't mobilize. And it's very, very safe. It's half a dose of normal anticoagulation. And it's been studied in older patients. And so we felt very good about introducing this just to reduce any risk of any venous thromboembolic event. So that is one thing that we are introducing moving forward in our protocol.
Interesting. Okay, great. All right. So we're talking about the fact that the latest results highlight both higher and lower dosing cohorts, excuse me.
Yeah.
How do the doses compare in efficacy, and what are you seeing in terms of the patient experience?
Sure. So it's too early to say for efficacy. So the way we're going to evaluate the doses is on target engagement. And by that, I mean levels of CSF progranulin. And so, as I mentioned before, we only have one data point of CSF progranulin for the lower dose. Based on that one data point, if the lower dose continues to look like this first data point, that will be enough. We will just use the lower dose for a registrational study. Now, again, we have to see what the next patients look like. Are they duplicating this early success? But what we've seen so far is looking promising for a lower dose. From a patient experience, there's no difference in patient experience. This is it's the dose. The actual dosing is very short. It's just a 2-minute infusion of you just there's a needle push for 2 minutes. So it's not really any difference in patient experience.
Great, great. Okay. Before we kind of sign off here, I would love to hear about certainly the next milestones that are expected and or any other additional outstanding questions you and your team have, both for clinical development, but also, what does this mean for the future of research into gene therapy for neurodegenerative diseases. What do you see coming out of PBFT02 impacting where we might be in 3 to 5 years?
Yeah, absolutely. Sure. So, our next milestones will be next year. We will share additional data from dose one and dose two, both efficacy data as well as safety data from our first patients who've been treated at the lower dose and with the prophylactic anticoagulation. We are also moving to an earlier patient population we're doing a protocol amendment right now to introduce the prophylactic anticoagulation, and at the same time, we're moving to treat earlier patients. I think that's one of the biggest things in neurodegenerative disease, not just for FTD, but for all neurodegenerative disease. The earlier you can treat patients, the better, because once neurodegeneration happens, it's very hard to actually replace what has already been lost.
Sure.
We're all trying to prevent this. And so one important thing, not only from our data, but from our peers in this space, from the data from our peers, and there's going to be some new data readouts at the end of this year in this FTD GRN space that are going to be very important. This data together, we are in the process of motivating patients and their providers to get genetically tested and to get into treatment as early as possible, because that's going to give them the best chance to get an intervention when it's going to matter. And I would say that's probably the biggest challenge for any genetic disease is in diagnosis, especially adult genetic diseases, because It's different from pediatrics. Obviously, there isn't screening for this. And so we need awareness. But awareness comes when there's successes. And so when you have scientific successes, when you have great data, when you have drugs approved, then patients get motivated, doctors get motivated, and that's what gets them into treatment earlier. And that is what is going to make the biggest difference in neurodegenerative disease.
Wonderful, wonderful. How about real quick before I let you go, patient advocacy groups being helpful in this regard? Are there any, even if they aren't specific patient advocacy groups, is that a big component to getting people tested sooner?
That is a huge component. There is a very large, well-organized group that we work with in the US, AFTD, the Association for Frontotemporal Dementia, that is very organized around all aspects of FTD, whether it's scientific research, whether it's patient engagement and screening. They have a fantastic registry. So we work with them quite a bit across all of these areas. And they are a absolutely wonderful resource for the community.
Okay, wonderful. As I suspected, and the reason I asked is because the patient advocacy groups across all of the various diseases, indications, what have you, that we write about and talk about, and the patient advocacy groups are absolutely a goldmine of information and helpful to the patients who have XYZ disease, whatever we're talking about. But so thank you for that information. And that wraps up this episode featuring Passage Bio's Dr. Will Chou. Will, thank you so much for your time. This was extremely interesting and informative, and I appreciate you and your time today.
All right. Thanks for having me, Erin. Take care. Bye-bye.
Thank you for tuning in, and be sure to share this episode with your peers and colleagues, and visit us at cellandgene.com, and we will talk to you soon.